फिर से
Good morning everyone, good morning, good morning. Good morning all. Okay, so friends, we were discussing about the cancer and we are left with a very little part of the cancer. We'll finish it and we will move on to the next topic immediately. Good morning everyone, good morning. Okay, so we understand what is the cancer, we understand the types of cancers, types of tumors and the characteristics of cancers. Especially we understand what is metastasis and angiogenesis. Later we understand that the cancer can be caused by the different agents like chemical carcinogens, radiations, and most importantly friends, biological agents. And I requested all of you to remember this particular table without fail. I hope everyone remember this particular table. Is it? Later what we have started? Friends, later we have started studying about the genetics of the cancer. Correct na? So when we studying the genetics of cancer, I'll quickly summarize in 1 minute. We have understood that there are more than 100 types of critical cancer genes were identified. What friends? There are more than 100 critical cancer genes were identified, but all those 100 critical cancer genes are categorized into two types, proto-oncogene as well as the tumor suppressor gene. I hope every one of you remember. We categorize it based on what? Based on gain of function mutation and at the same time dominant. I have told you to remember compulsory this. Proto-oncogenes are basically converting into the oncogene because of the gain of function, and it is a type of dominant mutation. And friends, when you look into the tumor suppressor gene, what you have to remember? It is basically loss of function. And most importantly friends, it is a kind of recessive mutation. I hope everyone remember, when I use a word dominant mutation, what you have to keep it in your mind. Even though one copy of the gene get mutated, that is sufficient, the entire cell can be converted into what friends? A tumorous. But here, if one copy of the gene got mutated, that is not at all sufficient to convert into tumor. If it wants to convert it into the tumor, and if it want to lose its function completely, friends, we have understood very clearly the two copies of the genes has to be mutated compulsory. That is what exactly we can call the underactivity mutation as well as the loss of function or a recessive function. Good morning, continue. Okay, so this is the last slide we have discussed, right? I have told you friends, don't get confused that there is a particular gene called as proto-oncogene is present in our body. Friends, in our body there is no gene as such proto-oncogene. Listen my statement. Any gene which is present in the body which is working normally, so that gene generally we will be calling it as the proto-oncogene only. Kindly don't get confused with that particular point. And the question is here, how does a proto-oncogene can be converted into oncogene? As I told you, there are multiple reasons are there, and one of the major reason is what? Gain of function. You people has to understand, you know very well, a gene already having a particular function, is it or not friends? A gene having a particular function, but the gene got an extra function which is not required actually. Because of that excessive function which it gained unnecessarily, The proto-oncogene actually converted into the oncogene. Kindly don't get confused with that particular point, friends. And every student listen. Here, all of you look into this. Proto-oncogene actually converted into the oncogene by a simplified mutation, deletion or point mutation. And the point is here, my friends, where does the deletion or point mutation occurred actually? It occurred in the coding sequence. See friends, understand what I am talking. Suppose example, if the mutation occurred in the non-coding region, there will be no much problem. Is it? Because non-coding region is not at all coding for the any kind of a protein. Hence there is no problem at all. But what you have to understand, the mutation was exclusively occur in the coding sequence, which is extremely important for the biosynthesis of a protein. And listen to me, I am not using the word protein is not getting synthesized. What is the word I am using is there will be a synthesis of a normal protein in a hyperactive condition. Hyperactive protein made in the normal amounts. What do you mean by the word hyperactive protein? Friends, listen to me. Hyperactive protein means even though there is no function of that particular protein, it starts showing its function. That is what exactly we can call the hyperactive protein, which basically leads to the tumor condition. And gene amplification. Friends, we know very well the gene should not undergo the process of amplification or duplication or replication in a many number of cycles. How many number of times the gene has to be amplified? It has to amplified only that many specific number of times only. But friends, here proto-oncogene, this gene, it got one function. What function it got? It got a function that through that function it is undergoing the amplification multiple times more than the normal. As a result, what is happening, friends? Same way, normal protein also greatly overproduced. Undoubtedly, it is converted into tumorous only. Next one, chromosome rearrangement. Every one of you listen to me very carefully, friends. Don't get confused here. The point. This is the gene, and the green color you can see, right, friends? This is the regulatory sequence. Okay, this is the gene, and this is the regulatory sequence. Kindly don't get confused with this point. Friends, understand, originally what is the function of a regulatory sequence? Regulatory sequence will control the expression of the gene. Is it? Whether the gene has to be expressed or not, or gene has to be expressed at what extent, the entire gene expression is under the control of this particular regulatory gene. So friends, that regulatory gene lost its ability to control this particular gene sequence. That's the point. Nearby regulatory sequences causes normal protein to be overproduced. Kindly do not get confused with that particular point. Because of that, what happens? This particular gene is going to synthesize the protein, which is either show the over response or their production only overproduction. This is what a kind of chromosomal rearrangement. And listen, this gene having a different function, the red color one, and this gene having the different function. But what happened now? The two genes which are having the two different function, both got fused. Both got fused. As a result, whatever the proteins that they are synthesizing, that proteins will become hyperactive completely. Kindly don't get confused with that. Two genes originally having two different function, but the two genes got fused with each other. Because of this sudden fusion, what happened? That genes start expressing more than the normal, which leads to production of a protein more than the normal. Kindly do not get confused with this particular point at all. And friends, I have clearly explained it to all of you. How does the cell surface receptor can cause the excessive proliferation? I hope you remember we have taken the proto-oncogenic receptor, and why do we call it as the proto-oncogenic receptor? Of course, there is no doubt they will be acting like a normal receptors only, no doubt about it. That's why I will be calling them as the proto-oncogenic receptors only, HER2 receptor and EGF receptor. Of course, to these particular receptors which will come and attaches, friends, undoubtedly growth factors will come and attaches. But every student, listen to me very carefully. We know they will be having a different kinds of domains. Quickly understand, it is having extracellular domain, transmembrane domain and cytosolic domain. We all people know that the three domains having three different functions. Is it, friends? Extracellular ligand binding domain. What is its function? Everyone listen. What is the extracellular extracellular domain function? Associating with the ligand. What is the function of a transmembrane domain? Transpanning the plasma membrane. And what is this particular, what we can say that intracellular domain, which is having intrinsic kinase activity. See, why I am specifying is domains, friends, they are individually working. Listen my statements, don't get confused. An example, suppose example, I have separated the cytosolic domain from the extracellular domain and the transmembrane domain. You people has to listen to me. The cytosolic domain is still active. It can show its activity. In the same way, I have separated the extracellular domain from the transmembrane domain and the cytosolic domain. Keep it in your mind, friends. The extracellular domain is still active because, listen friends, domains are the individual units. What do you mean by that? Which means domains can act individually also. At the same time, domains can act together also. Domain can act even in the combination also. Now there is a small mutation occur, and which area the mutation occur? The mutation occurred in the transmembrane region, and that too a very minute mutation of a point mutation. There will be a change in only one nucleotide. The point here, friends, even though there is a change in the one nucleotide, the change occurred at the coding region. What do you mean by the word coding region? Coding region means which are involved in the function. So you can see here transmembrane region, which is an important region which is responsible for transpanning the plasma membrane. So now listen to me very carefully. Just because of a single base substitution, valine converted into glutamine. As a result, what is happening because of this simplified mutation? Listen to me. Even in the absence of a ligand, even in the absence of a primary messenger, what is happening? The two inactive monomers become dimerized. The two inactive monomers become dimerized and they joined with each other. And friends, the cytosolic domain undergo the process of autophosphorylation. It can show the response. The cell starts growing and dividing uninterruptedly, which undoubtedly leads to the formation of the tumor. Same. Now you look into this particular EGF receptor. Then where the domain occur, where the mutation occur? Friends, understand, the mutation occurred in the inactive receptor tyrosine kinase. What do you mean by that? Mutation occurred in the cytosolic domain. Where the mutation occur? Mutation occur in the cytosolic domain. What do you call it? This particular mutation, constitutively active protein tyrosine kinase. We all people know very well, right? Whenever there is a presence of ligand, then only they will become dimerized. Their intrinsic kinase activity get activated. and they will do the process of autophosphorylation, and then only they generally pass the signals. We all people know that, but that is not happening here. That is not at all happening. What happening is the expression become continuous expression even in the absence of entire extracellular domain. Now you have to come to a conclusion. The mutation is not occurred in the extracellular domain, friends. Please kindly don't get confused. The mutation occurred in the cytosolic domain. So there will be no relation of whether there is a presence of the extracellular domain or not, because of the constitutive expression in the cytosolic domain. They will join together, and friends, they will undergo the autophosphorylation, and they can show the response continuously. So finally, what is the statement I can tell you? The statement I can give you is cell surface receptor can cause, everyone listen friends, can cause excessive proliferation. This is the point you have to remember. Friends, because of the excessive proliferation only, it is going to give rise to the tumor formation. Kindly do not get confused with that point. Okay, friends, this is about what? Proto-oncogenic receptor. Okay, now we have to read the most important for the exam point of view. That is what, friends? Tumor suppressor gene, which we can expect more commonly in our exam. If you look into your 2022 paper which you have written in the February exam, in the morning shift also we got the question from the PRB, and in the evening shift also we got the question from the PRB. Okay, I mean to say the way they have asked the question is a very similar way with a slight differences. Now you listen to me very carefully. Tumor suppressor gene. Everyone listen the definition of a tumor suppressor gene. Name itself very clearly indicating. Tumor suppressor gene is a type of a gene, listen to me, which is basically suppressing. What it is basically suppressing? It is suppressing the formation of a tumor. That's why we call it as the tumor suppressor gene. Friends, I want you people remember few points whichever I explained. If you remember that few points, we can apply that points for every single tumor suppressor gene, and we can understand it very easily. First point, what I want every student has to remember: tumor suppressor genes are not always active. First point. Tumor suppressor genes are not always active. Second point, they will be active upon there is a damage in the cell, or there is a oxygen deficiency, or excessive radiation. Only during these conditions only the inactive tumor suppressor gene is getting activated. Every student listen to me my words very carefully. The third point is the most important point. Friends, understand what I am talking about. Whenever the tumor suppressor gene get activated, the first point the tumor suppressor gene will do is stop the cell cycle. Stops cell cycle or cell division. Stops the cell division. First unique point. Then it will repair. It won't involved in the repairing. Don't get confused, my friends. It will activate the DNA repair mechanism. It is not involved in the DNA repair. Don't get confused. By chance, if whatever the damaged DNA that did not get repaired, immediately what happens, friends? Tumor suppressor gene will allow that particular cell to undergo the process of apoptosis, which we can call it as a programmed cell death. These are all the three major functions performed by the every single tumor suppressor gene, I can tell you, in a little different way, but the common mechanism of action is the same. So one point should be in your mind: every tumor suppressor gene is not active in our body every time. I am talking about every time. Whenever there is a particular mutation or whenever there is a damage, whenever the cells under very severe stress conditions like extreme deficiency of the oxygen, then the tumor suppressor gene concentration is becoming active. I'll tell you how their concentration increases. Ultimate function: first, it should not allow the cell to divide, it stop the cell division. It will immediately stimulate the DNA repair mechanisms, and DNA repair mechanisms try to repair that particular damage. If they are unable to repair that damage, we should not allow the cell to move to the next stage. If it moves to the next stage, undoubtedly it causes tumor. So that's why we have to do the process of apoptosis compulsory. So that also mediated by the tumor suppressor gene only. And friends, now we are gonna talk about every single tumor suppressor gene. Majorly you people has to remember for the exam point of view, P53 and PRP. These two are extremely important, friends. I want you people to concentrate on these two very, very much. Either in the part B or in the part C, these are all very common. Okay, so P53, P represents protein. Sometimes they can call phosphorylated also, but you always remember P represents protein. Okay, 53 is the molecular weight. Sometimes I told you na, instead of calling it as the protein 53, they will be calling it as the phosphoprotein 53. Anything is same. Friends, every student remember my words. Till today, I am talking about till today. Till today, one of the widely studied tumor suppressor gene in the field of the cancer biology. And I want every student has to listen my statements. One of the important statement: P53 is not active all the time. Not active always. Friends, listen my statements carefully, friends. Okay, and here I have told you one important line. What is the line I told you? Phosphoprotein 53. Every student listen my words. If the P53 protein wants to be active, it is important, it is compulsory, it is mandatory. The P53 has to be phosphorylated. Friends, please don't get confused with this particular word. Now you have to ask me, sir, when the P53 is getting phosphorylated? When the P53 is getting phosphorylated is whenever there is a damage. Whenever there is a damage occurred inside the cell, or there is a oxygen deficiency, or when the cell is continuously exposing to the radiation uninterruptedly, then what happens, friends? There will be a requirement of the P53. Then certain specific kinases will come and add the phosphate group to the P53. Now you have to listen to me. I told you that P53 is not active always. What is the reason? The reason, its concentration is extremely low, very low, its concentration. Now every student listen, friends. What is basically happening whenever the P53 get phosphorylated? Friends, listen to me. The concentration of the P53 is increasing very much, very high. The P53 concentration is increasing. The moment it got phosphorylated, its concentration increases, and it is doing three functions. What are the three? And one more very important one. In a given 4 statements, they will give one of the statement that P53 is a nuclear transcription factor. You know, most of the students will think that P53 is not a transcription factor, which is absolutely wrong statement. P53 is act as a nuclear transcription factor with a pro-apoptotic function. Please, friends, here in the cancer you have to remember a lot of statements, okay? Don't forget it. Pro-apoptotic function. Now you listen. When the P53 got phosphorylated, so its concentration is increased, it enter into the nucleus, and what it is going to do first, it is going to stop. It's going to stops the division. Common point, as I said, stop the division, activate DNA repair mechanisms. And the third and most important one, if they are unable to repair that mechanism, immediately, friends, they will stimulate the process of apoptosis. Kindly do not get confused with that particular point at all. Okay, so friends, every student listen to me very carefully, understand what exactly I am saying about. Oh, there is a power cut. Okay, now friends, listen to me. Now there is a damage of the DNA occur. When there is a damage of the DNA occur, everyone listen, the concentration of P53 rises. Correct na? Whenever there is a rising of the P53, every student listen, the cell is getting arrested at the G1 phase. You can see the picture very clearly. The cell is getting completely arrested at the G1 phase. And what happen? Everyone listen, they will start the mechanism of repair. They will start the mechanism of repair. If they are unable to do the process of a repair, immediately what is going to happen, friends? They will do the process of apoptosis. Then there will be no problem at all. But friends, now you people listen. Suppose example, there is a mutation in the P53 level. There is a mutation in the P53 level, and you have to understand. Of course, if there is a no mutation, there is a no problem. P53 arrest the cycle. But if there is a mutation in the P53, ORR, complete P53 is absent. Imagine there will be a no P53. What happens immediately? Immediately, friends, the cell is not getting arrested at the G1 phase. The cell undergo the uninterrupted cell division, undoubtedly, which leads to the formation of the tumor. Everyone look into the picture very carefully, friends. Look into this. See, this is the point you have to remember, friends. When the P53 is getting activated, this is very important. You should not consider that the P53 is not going to be released in the body every single time. Every student listen. During the damage of DNA, very low level of oxygen, that is hypoxia, very extreme deficiency of the nutrients inside the cell, oxidative stresses, hormonal imbalance, several impaired physiological processes. Impaired means irregular or imbalanced in a simple way. Whenever there is a impaired or imbalanced physiological processes will occur, then only, friends, the P53 concentration will become rises. And what is the statement I told you? Everyone listen. The first statement I told you, it will arrest the cell cycle. First one. Once it arrests the cell cycle, stimulate the DNA repair mechanism. If it is if the DNA repair mechanisms are unable to repair, every student look into the picture. It immediately allowed the process of apoptosis. Ferroptosis, apoptosis, necrosis, necroptosis. See, friends, what do you mean by the word ferroptosis? I will be explaining you in the apoptosis topic in the immunology. Ferroptosis means, friends, there is no doubt cell death only, but that particular cell death can be mediated by certain enzymes which will be requiring iron and sulfur as their cofactors. Once again listen. Ferroptosis means it's basically a cell death only. In that particular cellular death, certain enzymes are involved. Taken a best example of caspases. We all know very well. Caspases are the apoptotic proteins which are involved in the destruction of the cell. Is it? Friends, that particular caspases, if they want to be active and if they want to show their action, they will be requiring iron and sulfur as their cofactors. That's what we can call the ferroptosis. Same. See, apoptosis is the cell death. Necrosis is what, friends? Tissue death. That's all. And we are calling optosis. Here there is involvement of the iron and sulfur proteins. Now you just imagine a condition. P53 is not present. No P53. Now you only imagine, friends, these are all the conditions are happening. Now no P53. Do you think a cell will arrest? Never. Cell is never getting arrested at the cell cycle. The process will be continued and there will be no DNA repair occur, and friends, no death also occur. Undoubtedly it can form the